All right, welcome. So, this is the coronavirus spike protein. And I would imagine right now you're looking at this and saying, well, that's a lot of letters and a lot of colors and a lot of labels, and what does this all mean? My goal for the end of this, and your job for this week, is to be able to explain this one page back to me adequately with some detail. Because if you understand this, you understand the spike protein, and the spike protein is the sole basis for most of the vaccines and antibodies that we're going to be talking about, which is why we're going to focus on this one. It's just super important to the entire vaccine and antiviral development process. So let's go into this. So I'm going to show you this slide at the beginning, and I'm going to show you this slide at the end. At the beginning, it's not going to make any sense, and by the end, the goal is that this should actually make some sense to you. So let's try, shall we? First of all, just a little overview of what's going on here. So what we have is the protein, and it is shown as amino acids. So M is methionine, F is phenylalanine, V is valine, and so on. And so each one of these is a particular amino acid, and this is the order in which they appear on the spike. Now, there are a couple of interesting features on here that I've mapped with colors and highlighting. So there are a few places where the spike actually gets cut. It starts out as one long chain, and then it's actually going to get cut into 4 pieces before everything is over. So the first thing that happens is this blue bit up here, which is called a signal peptide, and there's our little signal peptide label. You can see I tried to match the color and shape, just sort of style. There is a signal peptide, and it's going to be cut off by an enzyme called signal peptidase, which is just the thing that cuts off signal peptides. So that's pretty neat, right? Signal peptides are there to direct traffic. If you have a signal peptide on a protein, it gets cut off. It's like somebody, punching your ticket when you go into the arena. And it wasn't about the ticket. The ticket is just a thing to get you in there. And so once the signal peptide is cut off, then this protein is marked for export. It is going to end up going toward the surface of the cell, and since this is on a virus, it's actually going to escape completely from the cell. But that's how the process starts. Next thing to consider, we have these things called domains. And all right, what the heck is a domain? A domain is a chunk of the protein that is going to fold up on its own, and it's either going to do a job, this entire chunk of the protein, in which case we call it like a functional domain, because it's a domain, part of the protein, that has a function, functional domain. Or we could call it a structural domain. If it just folds up into some sort of brick that seems kind of separate from the rest of the protein and would probably fold up into that brick shape even without the rest of the protein there, that would be called a structural domain. And so what we've got here are one of each, actually. So the first chunk here is highlighted in gray. So ignore these little purple ends for now. We'll deal with those later. But the gray chunk is called the N-terminal domain. So the beginning of the protein, think of it as like the engine of the train, is the N-terminus. It's where there's a free amino group, hence the N would be in the amino group, and that's why we call it the N-terminus. Down here at the other end, we have what we call the carboxyl terminus or the C-terminus, and that's just where there's a free carboxyl group on the end of the protein. So there you go, you're oriented. The N-terminal domain is the domain that comes first. And it sticks up kind of in the center of the spike like a little bump. We'll show you where that is in just a little bit. This is not a functional domain. It sits there, it has a structure. It folds up into a brick of some sort, but it doesn't seem to do anything directly involved with the way the spike carries out its business. So this is a structural domain, not a functional domain. And so there we go. We can knock off this entire thing. Throughout this domain, and actually throughout the entire protein, there are lots of places where there are sugar molecules that are joined onto the protein. And this is kind of a camouflage for the protein. And so we call this adding of sugar chains, we call it glycosylation, like glycose is like glucose. And so we're adding sugar molecules to the outside. This is something that just normally happens in the cell if you have a signal peptide. And it's pretty common. So everywhere there's a little N, this is a point at which one of these sugar chains is going to be attached. And the sugar chains can be tiny or they can be huge and branchy. There's a lot of ways that they look, and we'll actually go into exactly what these look like in a little bit. But so that's all the little purple ends knocked out. And so next domain, red domain. This is the one we really care about. So this is a region of the spike that sits up at the top of the protein, and this is the one that is actually solely responsible for sticking to the receptor, which is the protein on the cell that the virus is trying to infect. And we call this receptor binding protein, or receptor binding domain, the receptor binding domain. You'll see it abbreviated in papers as the RBD, but it just stands for receptor binding domain. That's it. And so it's everything here from the start of the red to the end of the red. And as you can see, there are 2 glycosylation sites within the receptor binding domain. Fantastic, right? Absolutely fantastic. Okay, then we have another domain down here that I didn't bother to label because, again, it's just a structural domain, not a functional domain, and we don't have to label all of them because whatever. If you can't see it and it doesn't do anything, why learn a name for it, right? Next, we have the first of 2 activating cleavage sites. The first cleavage site is cleaved by an enzyme called furin. And furin is another one of these proteases, or peptidase means the same thing as protease. A peptide is a small protein, a protein is a big protein, and so you can call it a protease if it cuts big things and a peptidase if it cuts little things, but you know what? They're the same enzyme, and they can usually cut both. It's I don't know, these are terms that people use, but these two are interchangeable, I would say. So, the furin cleavage site is going to recognize these particular amino acids. So starting here, R is arginine. So arginine, arginine, alanine, arginine, and then bam, furin will cut. It'll latch on to those arginine, arginine, alanine, arginine, and then it'll cut just before whatever comes next, which it actually prefers a serine, and we have a serine, and so that's where we cut. And so, yeah, physically, we have broken the chain here. Now, everything's still all folded up. This is just like cutting a little loop that's sticking out. Like if you had a whole plate of spaghetti, just cutting one noodle that was sticking out. Actually, no, if you had a whole plate of spaghetti, there was one giant noodle of spaghetti, cutting that noodle in one place, but the rest of the pile would stay pretty much intact. That's the idea here. Next little thing, D614 is one site where the protein is mutating right now. So this is a change that's been happening, and D is aspartic acid, and it's been turning into glycine, which is G. And so we don't know why this is happening because it's not in the receptor binding domain, so it's not going to directly affect how the protein works. It's kind of tucked away, I don't know, kind of halfway to the inside, down the little pocket. And honestly, other than the fact that this is a thing that is spreading in the world right now, it's hard to say exactly what this does. Eventually, we may know this. Actually, by the time you watch this, we may know this. Again, you got to stay on your toes with coronavirus. Okay, so furin, and then da-da-da-da-da-da, and then boom, we get to the really important one. So this is the cleavage site for a protease called either TMPRSS2, or there's a different protease called cathepsin type L, or there's another protease that was just discovered called TMPRSS4, TMPRSS4, and these are just the names of proteases, or I guess you can call them peptidases, although this is a big protein. Yeah, call it a protease. It's earned the title. Anyway, this is the cleavage site right here. It cuts after this arginine and before this serine, so it actually looks similar to the cleavage site here, except that the stuff that comes before the arginine-serine cut site is actually different. We have a PSKR instead of an RRAR, so there you go. Yeah. This is the cleavage that actually activates the spike properly. You can live with or without the furin cleavage. The virus actually does fine without it. It just helps make it a little bit easier for the virus to infect. But the TMPRSS2 or TMPRSS4 or cathepsin cleavage site is totally important, absolutely necessary for virus infectivity. After this, we come to the first of many little parts, and the rest of this, I'm going to do this in 2 parts. The second part, I'm going to show you each of these parts and what I mean there, and then at the end, then that's when you explain this back to me. So this thing is called the fusion peptide, and it actually is going to fold up into the shape of a little fish hook, It's got a little bend in it right in the middle, I think around where the D is maybe. And this little fish hook is going to embed itself into the membrane of the cell the virus is trying to enter. This is like a grappling hook that will allow the virus to sort of pull itself right up against the cell and crash the virus into the cell in a way that will actually lead to an infection. So that's what the fusion peptide is for. It's kind of the first thing that is going to be inserted into the host cell. First of many, many things we're going to do to the host cell. And if you see right here, the Tempress 2 or cathepsin or Tempress 4 cleavage site is right before the fusion peptide. The fusion peptide has to be free in order to do fusion. And so that's why this cleavage is super important for activating the virus. The virus does not work without it. All right, then we've got a couple other regions down here. So you got heptad repeat 1 in sort of lighter orange and heptad repeat 2 in kind of it's almost a rose gold kind of color I was going for there. And these two things are actually going to snap together. So heptad means group of 7, like hepta, the number 7 in Greek. And so these things are arranged with similar amino acids appearing about every 7. So here we've got Q, 1, 3, 4, 5, 6. Actually, N and Q are very similar. So the N is lined up with this NQ right here, the QN and the QN, which are lined up with this NQ right here, and so on. But there will also be, so these are fairly polar amino acids that are going to be lined up. In the middle, there are fairly hydrophobic amino acids that are also going to be lined up. And it doesn't look like it to the untrained eye, but what you got here is a repeating pattern of about every 7 amino acids. You will have amino acids with the same sort of properties. You won't necessarily have the same amino acid, but they will all be polar. Like here we have SS, and then about 7 later we have SS again. Then we have SR. The S's are quite polar. The R is both polar and a bit positively charged. And so it's just like that throughout the entire protein. These two are going to form 2 alpha helices, one here, and they're going to snap together because each alpha helix is going to have 1 hydrophobic side. Hydrophobic means parts of the protein that do not like to be around water. And so to hide from water, these two are going to clip together, they're going to hide together, which is kind of nice. When they do this, so the one is kind of attached to the fusion peptide, and the other one is attached to the transmembrane, which is this black thing, which is anchored in the actual virus. So this is going to bring the transmembrane very close to the fusion peptide, because these two things are going to snap together and they are kind of connected. So that is going to be the fusion motor, the thing that makes the virus actually work, the thing that makes the spike a fusion protein, a protein that is able to cause the virus to actually merge with, or fuse with is our sciencey word, the cell. In this region in between, we have what we call the histidine acid sensor, and I got a question mark there because we don't know which one is doing it, but probably some of these are doing it. Histidine is an amino acid that is normally not charged, so it's normally going to be hydrophobic. But when the pH drops down at about pH 5 to 5.5, histidine is going to pick up a proton, which is an H+ molecule, like hydrogen without its electron. And when it does that, it's not going to be neutral histidine anymore. It's going to be histidine with a positive charge. And so before, it was interacting with all the other hydrophobic amino acids, and now it is going to repel the heck out of those hydrophobic amino acids, causing this whole thing to kind of explode and change shape and then come back together. So we're going to be like a jack-in-the-box. We're going to start out in the coiled up, tightly coiled, energy-storing conformation, and then when we turn that last little crank and play, Pop Goes the Weasel or whatever stupid song they play, and when the acid binds to these histidines, then it is going to pop. And we're going to go through all those changes that result in this green bit ending up in the same membrane as this black bit. They're going to end up right together. Let's see. The red bit we know is important for the fusion process, but we don't know exactly why. We just know that if you mess it up, fusion doesn't work anymore. And then the only other bit to cover are these little orange C's down here at the bottom. Again, sort of a pumpkin-y, friendly color, yeah. Our pumpkin spice C's are actually anchor points, and you can see once again, these guys they're repeating about every 4 amino acids, 4 to 5, you're going to have a C. So from here to here is 4, to here is only 3, but to here is 4 again, and then to here is 3, and then to here is 3 or 4. And each of these is going to be the attachment point for a different kind of modification. We're actually going to add a fatty acid, just like the ones that make up the cell membrane, to some of these amino acids. So the C stands for cysteine, which is one of those 20 amino acids, and it happens to be the one that you can add fatty acids to. And so when there's a fatty acid attached to this, the fatty acid is going to try and hide in a membrane. And so we can turn this bit of protein, which would normally just be floating out there in the cytoplasm, into a membrane anchor by putting these little fatty acids on some of the cysteines. Now we think that about half of these end up getting occupied with a fatty acid. They don't all have to be occupied. There isn't even room to fit all the fatty acids on there. But, that's why they're in pairs. So you can do either this one or this one, and it'll be fine, either this one or this one, something like that. So on all the way down through it, and then boom, we're at the end of the protein. And that's the entire spike protein explained, kind of. Now, yeah, you try it. Or better yet, watch the next video and then you try it. That's a good thing to do.